Okay, so I had quite a few questions about the microscope, build a microscope lab. So I want to make a quick video to just talk a little bit about that and hopefully clarify some of the questions. So in a previous video, I talked about the first part of the microscope lab, which was using a single magnifying lens. You did some experiments with that. In this video, I want to talk more about the part 2, which was building a compound microscope. So if you scroll down the page, the Build a Microscope page, you'll find the compound microscope instructions here, where it says compound microscope. And it told you how to make the microscope. Then farther down, you have table 3, which is where you would have put your experimental data. So the microscope length, you were able to change the length since you had 2 tubes kind of inserted inside each other. And these are suggested distances of between the length of the microscope. The next column, you would put your experimental data, which was how far away the object was from the end of the microscope. And then the next column are just comments, what it looked like, if it was blurry, right side up, upside down, etc. Trial 2 was done if you had enough time for a teammate to look through the same microscope. He or she may have had to adjust the distance of the object to the end of the microscope slightly, but that's where you would put it. If you didn't have the opportunity to have a second person look through your microscope, then just disregard that part. The calculations for your microscope start in question 2, where you need to calculate the real image distance that was created from the objective lens of your compound microscope. And so to calculate the real image distance, you just use the equation 1 over f equals 1 over I plus 1 over O, like you did in the previous lab, in the previous part of the lab, part 1. If you would like a quick tutorial on how to do that, calculate using equation 1, I will do that in a separate video. Okay, so the main concepts for part 2 of the lab, which is making a compound microscope, are how to make a compound microscope, and particularly the placement of the lenses versus the focal points of both the objective and the eyepiece lens. Second, drawing a diagram illustrating the positions of the focal points, object and images, and lenses, actually. And finally, calculating the magnification of each lens and the overall magnification of the compound microscope. So when making the compound microscope, ideally, what you should have, of course, since it's compound, is 2 lenses shown here in this diagram. As objective lens, L1, and your eyepiece lens, L2. Then you want to place your object between 1 and 2 focal lengths away from your objective lens. So focal point is here for the objective lens. 2 times that is over here, so this object is in the correct position. When you do that, as you saw from the first part of this lab, you're going to make a real image somewhere on the opposite side of the objective lens from your object. So that's right here. That's the real image. You want the real image to be in a position between the focal point of the eyepiece lens, right here, and the actual eyepiece lens. So the real image should be in this range here someplace. And you could figure that out by knowing the distance from your first lens to your second lens. You know, that's your length of your microscope. And then knowing where the focal point of the eyepiece lens is and the location of the image. Make sure it's in between those 2 parts. If it's not, then you should change the length of your microscope so that that occurs. When you have that situation where the real image from your objective lens is between the eyepiece and the eyepiece focal lens, then you're going to create a third image, which is a virtual image, and that one is going to be magnified the most. So in short, here's where you need to place your lenses and your objects. The sample needs to be placed between 1 and 2 focal lengths of the objective lens, which is someplace right in this range here. If this is focal length of the objective lens, and this is 2 times that, so it should be right here. And then the real image that you calculate where that's going to land, that's a calculation, should be between the eyepiece lens and the focal point of the eyepiece lens. Okay, so that's how you should build your microscope. Drawing a diagram of your microscope could basically, at the simplest level, look basically like this here. You just want to, I didn't label this focal point of the eyepiece lens, focal point of the objective lens. That's missing. And then down here, farther down, you'll have the virtual image of the eyepiece lens. That would be way over here. And if you've done that, then you have the basics of what you need to have an optics diagram or diagram of your microscope as far as the optics go. I'll talk about the calculation of magnification of each lens and the overall magnification in the next video.